Andrew found his way into cellular agriculture quite early. He was already, I just learned, working together with Mark in 2013 and 2015, where he did internships at Maastricht University, or as Andrew just said, it was rather that Mark did an internship with him. He then did his PhD at the Tufts University together in the Kaplan lab, and is now working as a has now just founded together with 2 other people from the Kaplan lab a little startup that he might mention as well. There's 2 important things that you have to know about Andrew. The first one is that he makes really catchy names for the mediums that he develops. He might talk about this as well. And the second one is that he makes incredibly pretty slides. I hope I don't raise the expectations too much, but I'm pretty sure I don't. So yeah, with this, Andrew, stage is yours.
Awesome. Great. Thank you so much for the kind and hard to live up to introduction. I am going to take my phone and set a timer for myself to ding me after 20 minutes, because I can ramble. And I'm going to ramble today about cell line engineering for cultured meat. Thank you so much for the invitation to talk during this conference. I love this conference. It's very near and dear to my heart, so it's a real joy to be able to present to you all today. As Tobias mentioned, I did my PhD at Tufts University in David Kaplan's lab. I'm going to be presenting today mostly on work that I did during graduate school. And then currently, along with Natalie Rubio and John Yoon, we're starting a cell ag commercialization lab affiliated with Tufts to take the academic research that's happening at Tufts and moved it more towards industrial application. But today, we're going to talk about sort of these 5 things. And first, we're going to talk about genetic engineering as a powerful tool for cultivated meat. And why are we talking about it? Why do we need to talk about genetic engineering? It's a pretty simple answer. It's because cultured meat is really hard. It's really, really, really hard. And there are lots of headwinds that we're dealing with in cultured meat. And I've drawn them here for you, all of the headwinds of cultivated meat. So we're dealing with things that we've already heard about today. So we're dealing with toxic byproduct buildup. We're dealing with cell growth rates. We're dealing with metabolic efficiency. We're dealing with a lot of struggles. And this is us paddling upstream against the headwinds of cultured meat. But what do you know? We have this nice outboard motor sitting on our boat, ready to use. And if you look more closely, that outboard motor is genetic engineering. So what I'm going to try to sort of propose today is that we put that puppy in the water, we turn it on, and then it's off to the races. We're cruising upstream. We've got a fizzy little cocktail in our hands. My hair grows back. Tobias's hair grows back. And we're cruising. We're ripping. And so basically, to put that more scientifically than we're ripping, I think that cultured meat is so challenging that we really need to consider and develop every possible tool that we can to address the challenges. And so next, I'm going to talk about some examples from the Kaplan lab at Tufts, where we tried to do that, and we tried to engineer cells to address different challenges facing cultured meat. And so the first one I worked on a while ago, and it was genetically engineering cells to try to tune and enhance the nutrition of cultured meat products. And so this was motivated by the fact that lipid oxidation, which itself is associated with heme-bound iron in red and processed meat, lipid oxidation is associated with certain diseases that come from consuming a lot of red and processed meat, particularly colorectal cancer, where you get oxidation of this heme-bound iron, you get lipid oxidation, you get all these reactive oxygen species, which when you consume that food product and cook that food product and store that food product, those reactive oxygen species can interact with cells in your intestines, they can cause mutations, and those mutations can lead to cancer. And so we have these oxidation products, which are potentially harmful when consumed at high levels, but we also have chemicals that can help quench these reactive oxygen species. So specifically antioxidants. Antioxidants abound in all forms of life, and plants have a lot of antioxidants. And there's a class of really potent antioxidant called carotenoids, which are produced in plants and some bacteria, and which can quench those free radicals and potentially inhibit the onset of that disease progression that comes from eating red and processed meat. And so these carotenoids are not produced in animal cells normally, but they're produced in plants through this pathway where you have 3 enzymes. I'm not going to be able to use this well. You have 3 enzymes. You have SIRT-B, SIRT-I, and SIRT-Y. I'll get it off the screen. And those produce phytoene, lycopene, and beta-carotene. And you may have heard of beta-carotene before. It's the orange pigmented compound in carrots that turns them orange. It's what our bodies use to produce vitamin A, and it's also a really powerful antioxidant. So we asked the question, can we engineer these enzymes into bovine muscle stem cells and get production of these compounds? And so we engineered cells with SIRT-B, SIRT-I, and SIRT-Y. We saw that the cells maintained their phenotype, so our muscle cells still behaved like muscle cells, still showed the markers that we were expecting, and they were able to accumulate these carotenoids that they can't produce naturally. So SIRT-B, SIRT-I, and SIRT-Y can produce phytoene, lycopene, and beta-carotene, depending on which enzymes you're adding to the cells. And then because this, ultimately this is supposed to be a functional project, not just sort of a proof of the idea, we looked at whether this resulted in a reduction in lipid oxidation. We saw that when we stored and cooked pellets of these cells, the cells that were expressing the carotenoids and producing the carotenoids did indeed have reduced lipid oxidation. And then we also saw a color change in the cell pellet. Because this is an orange-colored compound, we saw that the cell pellets turned a little bit orange, which is cool, maybe not appealing from a food perspective, but I think it's pretty interesting. So this was kind of our example from a nutritional engineering standpoint, and I think this is just one example. There are tons of examples of how genetic engineering can be used in really cool ways to control and tune the nutrition of cultured meat products. So the next thing I'm going to talk about is immortalizing cells using genetic engineering. This is a topic that a lot of people are probably pretty familiar with, and we went about this in part, honestly, to just give us cells that we could use in the next experiment that I'm going to talk about. But to go through what we did a pretty standard immortalization process for these types of cells, where we expressed telomerase reverse transcriptase and cyclin-dependent kinase 4 in our bovine muscle stem cells. And what telomerase reverse transcriptase does is it lengthens the telomeres on the ends of the chromosomes of these cells. So those telomeres are protective regions that with every doubling are shortened slightly. And by the time they're fully shortened and removed, then you start with subsequent cell divisions. You get DNA damage, you get cell apoptosis, and the cells senesce. They refuse to grow anymore. And then cyclin-dependent kinase, what that one does is that basically pushes the cells from the G1 to the S phase of the cell cycle. So it provides this engine for cell division, telling the cells to continue dividing while the TERT enables them to do so. And this has been demonstrated before in muscle cell types from other species. And so we were pretty confident that it would work, and we were happy that it did work, and it worked quite well. So the black dots here are our primary bovine satellite cells. They senesce around the time we would expect, 40 to 60 doublings, that Hayflick limit. They start to really cease growing. Whereas our immortalized cells, they continue growing well past 100 doublings. And interestingly, because cell growth is kind of a self-selecting process for the faster growing cells, we see an acceleration of cell growth. And so by the time we're at 120 doublings here, our cells have a doubling time of about 15 hours, which we're pretty happy with, and that's quite fast. It's as fast as the C2, C12 cell line. And we see that the cells are able to maintain their phenotype. They express the markers of these stem cells that we would want them to express, and they're able to differentiate and fuse into multinucleated myotubes. And so I think that there are 2 reasons to do this immortalization. One is because from a bioprocess perspective, if you're always dealing with the same cells, then you can be sure that things are going to be really reliable. And the other is it allows you to do other engineering steps. With primary cells, say the carotenoids, we could engineer those to produce the carotenoids, but by the time that experiment was done, those cells were kaput. They were toast. They were pretty exhausted, and so it's not like we could. It was useful for the proof of principle, but it's not like we could use those cells then in a production process. And so to be able to engineer and continually upgrade your cells, you really need an immortalized population. And so we made one so that we could then approach this, because this is a really, really big headwind on our river is media cost. And so what we did was we engineered the cells to try to reduce that media cost. And for this, we started with 2 serum-free media that we'd previously made in the lab. One was called Beefy9, one was called Beefy-R. Tobias is very generous in saying that they're very clever. So Beefy9 is a 9-component media, hence why it's so clever, where there are a few components that really drive up the price and make it expensive. And most notably were albumin, recombinant albumin, and fibroblast growth factor 2. A project that we did that I'm not really going to focus on here was get rid of the need for recombinant albumin through the use of plant protein isolates, specifically rapeseed plant protein isolates. And so that's why Beefy9 became Beefy-R. And so in that case, we were able to eliminate this big cost chunk of the recombinant albumin in green, but we were left with this big cost chunk of the fibroblast growth factor in blue. And what fibroblast growth factor does for cells is it's a signaling molecule. So it exists in the media. It binds to cell surface receptors. In the case of FGF2, that's typically FGFR1, and it induces a number of signaling cascades. So I've highlighted 2 here. There's the P13K AKT pathway, which is turned on in the presence of FGF2, and you get an increase in proliferation and a decrease in cell fusion and differentiation. So the cells are told to divide, and they're told not to differentiate yet, because those are exclusive processes. And then also, this signaling phosphorylates the RAS protein, which turns on this RAF-MEK-ERK pathway, which also increases proliferation and holds off differentiation. And so if we were to, hypothetically, and we did it, so, but hypothetically, if we were to engineer our cells to express luciferase, that's a protein that isn't involved in FGF signaling at all. It just glows. So if we were to engineer the cells to express luciferase, then we would need exogenous FGF in the media for these cells to grow. There's some evidence that these cells might produce some of their own FGF, but it's not enough to tell the cells to grow. The cells still need FGF in the media. The other thing we wanted to try was if we instead engineered the cells to express FGF2 in an inducible promoter system, then maybe that FGF2 would be secreted from the cells and could either then self-signal, so autocrine signaling, or could signal neighbor cells, so paracrine signaling. We also wanted to look whether expression of a mutated form of this RAS protein was another way to go about this. So this is a mutation in the RAS protein that leads to its constitutive activity. So it acts as if it's always phosphorylated. So you get constant, you constantly have this RAF-MEK-ERK pathway being on and signaling. And then there's also some evidence that this mutated form of the RAS protein might increase the power of endogenous FGF signaling. And so then we also wanted to see, can we do both of these things at the same time and get added benefits from expressing both FGF2 and this mutated form of the RAS protein. We did this. It's no longer theory. And we got, I think, pretty exciting results. So to step through these, in the bottom bar here, these purple circles, what we're seeing is we're seeing control luciferase expressing cells in the absence of FGF2 in the media. The cells die as we would expect. The blue circles here are control luciferase expressing cells in the presence of exogenous FGF. The cells grow as we would expect. Then it gets pretty interesting. The pink and orangish triangles here are the cells either expressing just the FGF2 or just the mutated form of the RAS protein, and they're grown in the absence of exogenous FGF. And so those cells are now able to grow. And then the yellow line is when the cells are expressing both RAS and FGF2. The cells are able to grow comparably to our control cells in the presence of FGF2. And then this dotted yellow line is just that same cell system used in the Beefy-R media. So that's the cells where we're eliminating both recombinant albumin and FGF2 from the media. The big caveat here is that we see reduced differentiation in these engineered cells. And so I think that the sort of theory I currently have is, once we remove the doxycycline inducible factor, so that's what we use to turn on gene expression in these cells. Once we remove that factor from the media, there's still some in the cells. There's still some signaling happening. That signaling pathway doesn't turn off immediately. So I think what's happening is even though we're switching to differentiation media, there's probably still some of this FGF signaling happening, which as we know is, it reduces fusion of these cells. So I think that's why we're seeing this reduction in differentiation. I think it's a problem that can be addressed with controlling kinetics or time, or potentially looking into other inducible systems, or off versus on systems, things like that. But either way, we no longer have our FGF2 in the media. So with this, we've gone from a $75 media to I think it's about $18 per liter media, and this is all with pharma grade reagents that we could buy. So I think that the actual cost that could be achieved with this would be a lot lower. Okay, so check that one off the list, the examples. Hopefully everybody agrees that this is cool as a technology. But, spoiler, it's not just one technology. There are multiple ways to go about engineering cells, and they actually have a lot of implications about how it matters how you do it. So I want to talk about gene editing versus genetic modifications. So gene editing is the use of the use of no new non-native DNA. So when you gene edit, you're doing things that could theoretically happen through conventional breeding. So it could be a single base pair edit. You could mutate a gene to upregulate it or turn it off or something like that. Or you can add another copy of a native gene, a cow gene, to a different part of the genome, say to a really highly active promoter region, or with a really highly active constitutive promoter. And that would kind of be treated as being all cow DNA, so these mutations and shufflings. Whereas genetic modification is when you have transgenes, so genes from another species that's not sexually compatible with the species that you're inserting into. And so that would be, for example, taking a beta-carotene synthesizing gene from a carrot, putting it in a cow cell, and some people have issues with that, which I'll get into specifically. So those are kind of our two options. And it's important because they're regulated very differently depending on what jurisdiction you're in. So here, all the countries in the dark green are places where they don't treat a gene-edited crop. This is typically considered in the world of crops, but I think that the translation to cultured meat is a pretty easy one. So a gene-edited crop is not regulated as if it's a GMO. It's basically regulated more closely to just as the conventional food product. The light green and yellow areas are countries where there's sort of ongoing legislative conversation about what's going to happen. And then the red countries are where a gene-edited product is regulated as a genetically modified organism. So in these green areas, if you have the option to take the gene-edited route, then your regulatory hurdle is likely to be pretty low. In the red countries, no matter what route you take, the regulatory hurdle is pretty high. Obviously, we're here in this red spot. So in the context of this conference, it really matters, that distinction. But also, interestingly, I think things might change. I'm not a European regulatory expert. We're going to have a panel later with a European regulatory expert, so we'll definitely talk about this later. But just to kind of prime that, in July of this year, the European Commission released a proposal or a report kind of proposing that the EU take a different approach where they start differentiating these two technologies. They use the term new genomic techniques rather than gene-edited, but the proposal is to start regulating new genomic techniques similar to conventionally bred crops and food products versus regulating them as genetically modified organisms. So there are some other practical considerations. The regulatory one is obviously a really important practical consideration, which we talked about just now. But there are some other practical considerations when one thinks of engineering cells that I just think it's interesting to talk about. So the first, obviously, is consumers. I definitely realize that a lot of consumers have qualms with gene editing, with GMOs. We can sit here and talk about the difference between gene-edited and GMOs all the time, and my aunt she still thinks they're all GMOs, and she's against them. But there is some differentiation amongst consumers between these different technologies that are used. So on the left here was a study, a survey study looking at whether consumers were opposed or in favor of food products that were produced with genetic modifications or with CRISPR technology, so a gene editing technology. And in the USA, Belgium, and France, there was an increase in acceptance when that CRISPR technology was used, as opposed to sort of a transgenic GMO product. It also depends who makes these. This was a study in the US where basically consumer opposition was studied to GMOs, and depending on who produced the GMOs. So over here on the far left, so basically the red line is percent opposing. And so over here on the far left, you have much more people opposing if it's Monsanto, right? Monsanto, I think, has not done a great job of shepherding the PR of their GMO foods. So it's not really surprising. Interestingly, for this conference, so a Silicon Valley startup kind of has also a pretty unfavorable perception from consumers. I think a lot of consumers don't like the techies. And then over here, there's a lot more acceptance when it's a nonprofit science institution, a university, or in this case, the US Department of Agriculture. I have 5 minutes left. Okay, so I'll go to the next slide. So that's all to say, I think it's just, all of these are meant to just sort of be considerations. I don't want to be prescriptive at all and say, Silicon Valley startups shouldn't do GMO work. Obviously they should. But it's important to kind of consider all of these nuances, because I think the debate is typically GMO, non-GMO, when really it's much more nuanced. And so then the final thing I want to talk about from just a practical standpoint is, as these technologies are aimed towards commercial application, there are some real sort of IP considerations to make. So CRISPR is a super, super, super cool technology that is incredibly powerful for research, incredibly new, incredibly exciting, and incredibly heavily patented because it's so new. I think that, again, obviously, if CRISPR is the way to get to the target that you want, then use it by all means, but you'll just have to, if you're producing a product, you'll have to license the technology. So that's just a cost you have to consider. And there are some older techniques that I think people often discount because they aren't that flashy new CRISPR technique, but they have some real advantages. So I just think this is pretty interesting and timely for this talk. So the Phi-C3 recombinase system was patented for the first time on October 14th in 2003. Patents last 20 years. That patent expired 2 days ago. And so what that is it's a way to have any insertion, but in the example of gene editing, it's a way to have a cisgene insertion. So basically, you could have a cisgene on a plasmid that has an ATTB site, and then many organisms have pseudo-ATTP sites within them at known locations. And the Phi-C3 integrase will basically cut and paste this genetic cargo into that region without, if you design your plasmids well, it'll do it without adding any new genomic material. So it'll all be, it'll still be all cow, you'll be all clear, and it'll do it at known locations in the genome. So from a, cell line integrity, regulatory perspective, basically just knowing exactly what you're doing, I think this is a pretty cool technique that's pretty free to use. I think it's just sort of a consideration that I find pretty interesting. And so genetic engineering, some examples, hopefully, that seemed useful, and some considerations about the nuance and how to sort of go about it from a practical standpoint. And then once we've thought through all that, I think the possibilities are really, really, asymptotically close to endless. There have been many examples in other fields. So CHO cell line engineering is an old field, much older than cultured meat, and they do a lot of really, really cool stuff. And I think what's interesting and exciting is you find so many papers where people have engineered CHO cells to increase the production of a recombinant protein, and they say, oh, we made X, Y, or Z mutation, and we didn't see an increase in recombinant protein production, so we kind of discarded this path of inquiry. But then you look at the growth data, and oh, well, the cells grew faster, and the cells grew to a higher density. It just didn't result in an increase in recombinant protein. So I think that there's so many opportunities that engineering strategies that haven't been fit for purpose for other fields, but which will be fit for purpose for cultured meat. And so there's kind of this wealth of research that we can still go back on and look for exciting opportunities. And then lots of cultured meat companies are working on patenting work in genetic engineering. So this is just a few. I know that I'm missing a ton of them, but just to kind of sprinkle in ideas about where this can be used. So that there's work on decreasing toxic waste buildup. There's work on decreasing adherence, lack of adherence induced death, so anoikis in cells. And then there's some cool work on optogenetic control of cell signaling. So lots of really exciting paths to continue down. And once we do, we're off to the races. So to conclude, before questions, I think to summarize, we just need to consider this technique. It's really, really powerful, and it obviously won't work in every instance, and there are very real reasons, regulatory, consumer, scientific, to be conscientious about our use of it. But I think it would be irresponsible not to at least sort of consider it. There were some examples from our work that hopefully sort of help show the power of this. And then there are all these other interesting considerations, which I find very interesting. And I hope you did too. And with that, I would just want to thank the Tufts University Center for Cellular Agriculture, which is sort of the umbrella that holds a lot of Tufts University's cellular agriculture work. Like I said, I did this work in the Kaplan Lab, so huge thank you to the Kaplan Lab and to the many, many undergrads who were huge in this work. Thank you so much to New Harvest, who funded my research. I'm enormously grateful. And then to the other lab funders who have continued to fund the Kaplan Lab. And thank you very much, and I would love to take questions.
Thanks so much. Really great talk. I'm sure there will be plenty of questions, and I already see a few hands raising here. Elliot, maybe? Oh, there's some. Okay.
Yeah, thanks, Andrew. So in humans and other model organisms, there's some safe harbor sites in the genome that are commonly used. But to my knowledge, these haven't really been characterized in a lot of the species that we use for cultivated meat, especially like seafood and things like that. So I'm wondering your thoughts on, like, from a practical perspective, do you think that's going to be of high utility? And also from a regulatory perspective, is that something that people will consider for like stability and things like that?
That's a great question. Yeah, so I think, you're totally right. So in some species, like the bovine Rosa 26 or AVS, so these are sites where it's known that you can put new genomic material without disrupting cell behavior, and it's pretty stably expressed. I think they're super awesome and useful spots to put DNA. So I think that, like, finding out where, sort of analogous things in other species is super, super useful. I think that I would say that, I don't think that it necessarily needs to be these sort of like canonical Rosa AVS1 safe harbor sites that we think of as sort of our only options. I think it'd be unfair to set ourselves the bar. I think we can almost as a field define that bar with regulators of what are acceptable regions to put this. I think, like with the 5C31 recombinase, the important thing is like knowing where it is and being able to track stability and things like that. So I'd say, we know that those are useful sites, but they're useful in the fact that we know them, we trust them, and they're stable. And so I imagine that there are lots of other ways to get to those same goals, not without necessarily saying like, oh, the only way to produce a safe culture meat product is by use of these safe harbor sites. If that answers the question.
Yeah, thanks.
Yeah, thanks.
Any more questions? Here. you can also pass the microphone to Alberto, to.
Wonderful talk. I'm just curious, looking at the differentiation results that you had, was that in just the double mutation, or was it in the single mutations as well?
That was in just the double mutation. We did it for the single mutations as well. We saw worse differentiation in the double mutation than in the single mutations, but we saw those ones were still also worse than our control. Yeah, and that was sort of the one caveat that I had time for. We also see all of the engineered cells in that FGF2 work have a lower doubling time than our non-engineered controls. So I think like the system needs a lot of tuning and work. So it's definitely not ready for prime time, but I think hopefully an interesting starting spot. Thanks.
There are some questions back there. You can pass on.
Thank you. When you immortalize your bovine cells, did you use the TERT from the isogenic sequence or the different species sequence?
Yeah, so we used bovine TERT and bovine CDK4. They were on the CMV promoter. So once we did that promoter, they're in the realm of being transgenic, not cisgenic. Those would definitely be a GMO. But I think I kind of always kick myself about this because it would have been really easy to just use an endogenous promoter. And,
okay.
Yeah.
One more question is what is your opinion about the spontaneous differentiation? Somebody's called natural differentiation versus this induced or GM.
Yeah, definitely. So I think spontaneous immortalization is an awesome way to go because you then don't even have to start having this conversation. There's an interesting paper that came out pretty recently that looked at spontaneous immortalization rates in cells from animals of different sizes, and they found that the larger the animal, the much less likely you were to have a spontaneous immortalization event. And so the theory that paper kind of proposed was, these animals grow to really large size, and so they can't afford to have spontaneous immortalization events, whereas smaller animals, their DNA repair mechanisms maybe don't have to be quite as good. So I could see it sort of being differentiated where for a cow, spontaneous immortalization events are really rare. I've not heard.
Chicken.
Yeah, as opposed to chicken or fish where they're more common.
Okay, I think in respect to time, I'm apologizing to everyone who would want to ask questions, but I'm sure you can ask Andrew later after this panel. But let's give Andrew another round of applause.
Thank you very much.
Thanks for this really great talk.
Thank you so much.